I don't have any reference here, Mr. Ronaldo Mukhari. I hope I pronounced it correctly. And we continue, we're going to talk about long-term high-fat diet, which is interesting in the aged vervet monkeys, and we're looking forward to your presentation.
Thank you for the introduction. So once again, greetings to everyone on the floor. As introduced, my name is Ronaldo Mukhari, and I'm here to present my study, which is titled The Effect of Long-Term High-Fat Diet Feeding on Intestinal Health of Aged Vervet Monkeys. So westernized diets are a category of unhealthy, calorie-dense foods that are enriched in simple sugars and saturated fats, but lack in fibers, vitamins, and minerals. And the consumption of westernized diets such as a high-fat diet has increased in middle and high-income countries. And unfortunately, these diets dysregulate intestinal barrier components, and this may lead to subclinical systemic and tissue inflammation that is implicated in the development of diabetes. Now, to further outline this, a healthy gut or a healthy intestinal barrier is distinguished by its ability to prevent the translocation of luminal antigens across the intestinal epithelium and eventually into systemic circulation. So as such, this environment must be tightly regulated, and components of the barrier that are responsible for this include immune cells such as plasma B cells, and these are involved in the release of an important immunoglobulin IgA. And so some of the information that I'm going to be sharing in the background is going to be a refresher since Dr. Sylvia has outlined a few. So another essential component besides IgA is tight junction proteins. So these proteins are responsible for forming a physical barrier across the intestinal epithelium such that they prevent unwanted or increased intestinal permeability. So altogether, deregulation of these components, among others, including changes in the morphology of the intestines because of a high-fat diet, leads to the translocation of unwanted molecules across the epithelium and eventually into systemic circulation as a result of increased intestinal permeability. Now, the translocation of these unwanted molecules, an example is the bacterial endotoxin LPS. Now, to further outline this, the accumulation of LPS in blood is indicative of metabolic endotoxemia. And metabolic endotoxemia is a condition that is characterized by the recruitment of molecules such as cluster of differentiation 14, CD14, and LPS binding protein LBP. Now, formation of this complex triggers or activates the toll-like receptor 4 signaling pathway, which then now induces the release of inflammatory molecules such as nuclear factor kappa B and mitogen-activated protein kinases, which are implicated in low-grade inflammation, thus diabetes. Now, on the other hand, a high-fat diet may dysregulate mucosal immune cell populations or IgA-positive cells, which, according to literature, the dysregulation of IgA-positive cells has been associated with impaired glucose metabolism. Thus, this may potentially facilitate the development of diabetes. So, while it is evident in literature that a high-fat diet can exert both local and systemic inflammation that is associated with a high-fat diet, the the mechanisms of a high-fat diet-associated inflammation are complex and need further elucidation. And venturing into this may lead to the identification of diagnostic and therapeutic targets to prevent or ameliorate the diabetes and its associated complications. And one of the ways to study the pathophysiology of diabetes is the use of non-human primates or monkeys. And they are considered a good animal model because they share similar clinical and pathological features of diabetes with humans. So hence it suffices to use the model to investigate the effects of diet on intestinal health and its role in the development of diabetes. This then now brings me to the aim of my study, which was to investigate the effect of long-term feeding of a westernized high-fat diet on the intestinal barrier function and inflammation in intestinal tissues of vivid monkeys. The objectives of the study were to assess the effect of a westernized high-fat diet on liver function, glucose and lipid metabolism. And then we also assessed biomarkers of intestinal barrier dysfunction, LPS and CD14 using ELISA. And then we also looked at the morphology of the intestines using hematoxylin and using staining. And lastly, the expression of tight junction proteins and mitogen activated protein kinases was assessed using immunohistochemistry and Western blot. So in this study, we used tissues and serum samples that were previously collected at the Primate Unit and Delft Animal Center of the South African Medical Research Council. And so in this study, a total of 25 vivid monkeys were used. 12 of them were fed a maize diet, and 13 of them were fed a high-fat diet for a period of approximately 15 years. And so after the animals reached an advanced age, they were euthanized, blood samples and tissues were collected and stored for later use. And ethical approval for this study was acquired from the SAMRC. And then we went on to assess liver function, glucose, and lipid metabolism by evaluating markers such as AST, ALT, fasting blood glucose, total cholesterol, triglycerides, LDL, and HDL cholesterol. And then in the serum samples of the monkeys, we assessed markers of intestinal barrier dysfunction, which are LPS or which are LPS binding protein and cluster of differentiation 14. And then we used hematoxylin and eosin staining to assess the morphology of the intestines, where in the ileum we measured villus length, and in the colon we measured crypt depth. And during this technique, we excluded the duodenum and jejunum sections since they were too digested or autolyzed during a sample collection and processing. We also used immunohistochemistry to assess the expression of a tight junction protein, which is involved in maintaining integrity of the intestinal epithelium. Thus, it plays a role in preventing unwanted or increased intestinal permeability. And we also used immunohistochemistry to assess the population of IgA positive cells in intestinal tissues. And lastly, we assessed the expression of an example of mitogen activated protein kinases, which is extracellular regulated kinase, And these have been reported to play a role in the synthesis of inflammatory mediators. And for statistical analysis, our data was represented as mean plus standard deviation, and statistical differences between the two diet groups was calculated using graph-paired prism by a two-way ANOVA and unpaired t-test. And the p-value less than or equal to 0.05 was considered significant. So in our data, when assessing the effects of a high-fat diet on liver health, when looking at figure A, it was evident that ALT levels were higher in the high-fat diet group when compared to the maize diet group. And also, ALT levels were higher in females when compared to males, regardless of diet. And we also observed an interaction effect where females that were fed a high-fat diet had higher ALT levels when compared to males fed a high-fat diet as well. And then looking at another marker of liver health, AST, from our data, it was clear that AST levels were increased in females when compared to males, regardless of diet. And there was also an interaction effect where females that were fed a high-fat diet had higher AST levels when compared to males fed a high-fat diet. And then we also assessed glycemic and lipid parameters. And from the data, we can see that there was an increase in fasting blood glucose levels in females when compared to males, regardless of diet. And we also found that diet did not have an effect on the levels of triglycerides. And then when we assessed the levels of total cholesterol, it was evident that the high-fat diet group had high levels of total cholesterol when compared to subjects that were fed a maize diet. And then we also observed that there was an interaction effect where females fed a high-fat diet had lower levels of total cholesterol when compared to males fed a high-fat diet. And then the diet did not have an effect on HDL cholesterol levels. However, we did find differences in LDL cholesterol levels where the monkeys that were fed a high-fat diet had higher levels of LDL cholesterol when compared to animals fed a maize diet. And also, LDL cholesterol levels were higher in males when compared to the females, regardless of diet. And we also observed an interaction effect where the females that were fed a high-fat diet had lower LDL cholesterol when compared to males fed a high-fat diet. And then when assessing the effect of a high-fat diet on intestinal barrier dysfunction, we found that diet did not have an effect on LBP levels. However, it did have an effect on CD14 levels, where animals that were fed a high-fat diet had higher levels of CD14 compared to those fed a maize diet. And we also observed that CD14 levels were high in males when compared to females, regardless of the diet. And then, so before I proceed on to the next set of results I'm about to present, I'd like to outline that for the next set of results, we had a lower sample size compared to the results I've presented thus far. And also, in the next set of results, what we did is that we assessed the effect of a high-fat diet on intestinal morphology and the expression of specific proteins in intestinal tissues. And this was for us to understand the underlying mechanisms. So firstly, the images were acquired through hematoxylin and eosin staining, and they were captured from the ileum and the colon. And from the images, we assessed villus length and crypt depth. And when looking at the graph, we can see that diet did not have an effect on villus length in the ileum nor crypt depth in the colon. And then these images now depict the presence of IgA positive cells in all of the sections of the intestines. And from these images, we then quantified area that is positive for IgA. And from our data, we found that a high-fat diet increased the population of IgA positive cells in the duodenum when compared to animals that were fed a maize diet. However, this was not the case for the jejunum, ileum, and colon sections. And then we also assessed the expression of a tight junction protein occludin, and as you can see in the pictures, it's denoted or is depicted by the green fluorescence. So from the images, we then quantified its expression, which we represented as mean fluorescence intensity, and we observed that diet did not have an effect on the expression of occludin in the ileum and the colon between both diet groups. And then we assessed the expression of extracellular regulated kinases, ERK, and these have been reported to play a role in the synthesis of inflammatory mediators. And from our data, we found that diet did not have an effect on the expression of this protein in the ileum and the colon. So this then now brings me to the conclusion of our study, where it was evident that long-term consumption of a high-fat diet in vervet monkeys resulted in hypercholesteremia, and this may increase the risk of cardiovascular disease. And the long-term consumption of a high-fat diet also resulted in liver and intestinal barrier dysfunction, which have been linked to the development of diabetes. So overall, this study suggests a potential link between the consumption of a high-fat diet, impaired gut and liver function, as well as an increased risk of both cardiovascular and metabolic diseases. So as such, future studies should seek to understand the underlying mechanisms by which a high-fat diet disrupts the intestines and contributes to the development of chronic health conditions. And so what we'll do in the future, following an increase in the sample size of the study, we are going to do further assessment of intestinal morphology using hematoxylin and eosin staining. And we'll also stain more tight junction proteins besides occludin using immunohistochemistry, and we'll also use Western blot to assess expression of these proteins. And these are the references I used for my presentation. I would like to acknowledge the following people, institutions, and organizations. And this is the SAMRC transformation slide. I thank you for your time and attention.
Well, thank you for your very nice presentation. Very good. And I think there's room for 2 questions. First question over here. Nadina.
Yeah. Thank you very much for an inspiring presentation. I have 3 minor questions. First of all, did the monkeys under high-fat diet gain weight? Then second one, 15 years of feeding high-fat diet, is this long? Because I have to say I'm very ignorant for monkey models, so I have no assumption if monkeys get 60 years of age or if 15 years of age is an old monkey. And the third one is, what about the heart? How did the heart look like?
Okay, to answer your first question, the monkeys did not, there were no significant differences between the weights in both, between the diet groups. And so with regards to the age, I'd say that an average lifespan of a monkey is around 20-something years. So 15 years, it's a long time. And your third question,
it's about the heart.
Oh,
so
the heart.
Do you have an insight about the heart? Because you mentioned we know that high cholesterol can have a cardiovascular risk, but how did the heart look like in these monkeys?
I didn't get to look at the data. I'm not sure if it's available, but it's something that if it is available, we'll do further analysis and actually get an in-depth of what overall effect did the diet have.
Yes, thank you very much.
Thank you.
Any other question? Yes, over the right.
So as far as the calories consumed daily for the monkeys, were they on an ad libitum diet or was it actually on a set schedule?
It was ad libitum, and it was ad libitum, yeah.
Any other questions? Because in that case, I also would like to. Is it working?
I don't think it's on.
It's working now. I also have a different question. And of course, we had a hard question, but for me, you had a nervous question, okay? Because I was a little bit surprised when you take out the intestine itself is enriched on the surface, on the inner surface, with the so-called supraepidermal plexus, with a large number of enterochromaffin cells, so basically nervous tissue. Didn't you look at all to nervous tissue? Because one of the things, maybe, I'm just giving you a hint, the enterochromaffin cells produce serotonin. And this lack of serotonin by a high-fat diet could induce leakage. So I would ask, or maybe suggest, maybe in your beautiful immunocytochemistry work, and you can have it from our lab for free, some antibodies for serotonin, and you can look to the supraepidermal plexus and the changes between high and low-fat diet.
Yeah.
But how do you feel about this?
Yeah, that's very interesting, and yeah, I agree. Because there is an established link between the gut and also the brain. So yes, and yeah, definitely we look at it's something to look at. The levels of serotonin, yes.
And basically, you're a PhD student.
Yes.
So that means in the future you have to do a thesis defense, which we would do in my country. So I'll ask you a question about this. And I'm going back to Nadina. I was a little bit surprised that you told me 15 years of high-fat diet does not result in any increase in body weight. I'm stunned. How is that possible? Do you have a different diet here in Africa than we have than in we have?
I'm not sure. Perhaps it could be maybe the percentage of dietary fat, because the composition, the high-fat diet, the high-fat diet group had 40%. The percentage of fat was 40% within the diet. So it could be that, I don't know. It could be that perhaps we needed to administer a higher percentage, and then that could have resulted in changes in weight.
Okay.
Yeah.
Good. Let's stop here for now, otherwise we get too much. Thank you very much.
Thank you.